Go ahead. Oh, just
Okay.
do me one thing. Do me a favor, Swara. Can you get rid of that meet.google.com is sharing your thing? Yeah, just get rid of that.
Okay.
Okay. Yeah, it's hard now.
Okay. Okay, so this is SafeCRISPR AI.
Okay, interesting.
So my project is a simulation of CRISPR. And so CRISPR is a gene editing technology that basically allows the modification of DNA. And so I built SafeCRISPR AI as a simulation, which is designed to improve the safety of CRISPR by predicting and reducing the off-target DNA modifications before scientists have to go into the wet lab. And so it combines many different models and analyzes a lot of data sets in order to help scientists make more informed decisions.
Interesting.
Yes. So the problem is, so CRISPR is a very advanced technology, and yes, it is extremely helpful when getting rid of dangerous mutations and transforming modern medicine and slowing down cancer. But sometimes CRISPR can unintentionally cut DNA at the wrong locations. And so this can introduce harmful mutations, it can reduce the effectiveness of the treatment and create a lot of safety concerns. So even though existing prediction tools struggle with the accuracy or efficiency, I chose to make a simulation to choose the safest guide RNAs or DNA sequences before, like as a support system before conducting laboratory experiments. So gene editing is already being explored in many ways in the real world with many diseases like Huntington's, sickle cell, blindness, and a lot more. And so this makes prediction accuracy a very, very important factor. And so if even a single unintended DNA edit could have serious consequences. And this means that we need to have a good, accurate support system before wet lab testing begins.
Okay. Yeah.
So SafeCRISPR AI provides, it's a simulation, and it's a simulation prediction platform where researchers can upload or input DNA sequences, chromosome locuses, the species, and other information in order for the AI system to evaluate potential off-target effects using many models like transformers. And it generates a safety score and analyzes all the information to predict likely off-target and ranks guide RNAs based on the risk. And so it supports the lab results.
Okay. All right. Please do show me the demonstration. I'm curious about this.
Okay. So this is my landing page here.
Sorry, could you just make this full screen? Press F11.
Sorry.
Press F11. Make this full screen.
Okay.
Function D F11.
Uh,
Okay, it's fine. It's fine if you can't. We'll do it later. Okay, just keep going. Sorry, I threw off your flow there. Right? Can you please continue?
No, it's okay. So this is my landing page. It has the features, compares models, and basically gives the reader an overview of what the website does. So I will go to the platform now. All right, so here the reader can enter a lot of different information, and usually a researcher is going to be putting this in. So this will be like the name of the study. This will be the species. Right now it's only 3, Homo sapiens, which are humans, and then of course most. Yeah, and then this is the chromosome that they're working on or editing, and then the coordinate that they will take from their machines. And then, of course, they will input their a specific DNA sequence that they have to edit, and then they're going to input the RNA sequences that they want to add to the DNA. So what they're trying to add with CRISPR.
Interesting. Okay.
Yeah. So here you can press execute model pipeline. Of course, this is a simulation. So yes, it has realistic data sets, but in order to make this more accurate, I would need to have a lot more data, which I will work on in the future. And so here it will give us a lot of analytics. So it gives us the top performing RNA guide out of the 2, or they can add even more, which I will show you. They calculate the risk score. So this one is pretty good. Here we can see the quality, seed integrity, and then it tells you, like, all these numbers are out of 100 and tells you what each means. Of course, the researchers will already have an idea of this. And it tells you the confidence of all our models combined. So it won't be 100%, but it will be pretty close to it. And then there's a different graph based on the input here. This, you can explain the graph in case they're unclear. So like for this chromosome graph, I clicked on explain graph and it tells you the visualization provides an automated model of double strand fragmentation. Yeah.
Got it.
And then it tells you the fidelity score. So as you can see, the first RNA guide was pretty strong. It is tier 1, but the second one is very, very risky. It's tier 3. Here we have a sequence alignment matrix. So I'll show you an example of adding more here. I'm going to change the sequence just so you can see how it's not hard-coded, and I'll add one more here. Right, so as you can see, the percentage changed, and this one seems much, much more risky. It gives you a critical caution warning. This is a heat map that displays the risk intensity across the cleavage loci. As I was saying before, the sequence alignment matrix, it's just a diagram or a model of which letters and nucleotide bases match and which one don't. So as you can see here, there are very few that match.
Right? Yeah, got it.
And these are still here. And then the ensemble frameworks are just comparing all our models. I use 4 in here.
All right, pause. Okay, go back to the models actually.
Okay.
I want to show the models, right? Okay. I want to ask about them. Actually.
Yeah.
Is it a bit more down?
Oh,
No,
these?
show the 4 models, right? That you're using.
Oh, these.
All right, there you go. Yeah. Okay, so using DNABERT, using deep CNN, the random forest, and XGBoost. Huh? Okay, so like, are you using all 4 of them like parallelly, or like one after the other? How exactly is this going on?
It's analyzed. Yes, it's analyzed by 4 different models, like simultaneously, and then each model will predict the probability that the guide will produce